Good morning. This is me again with a recording for the lecture on chapter 10 photosynthesis. It is a good idea to listen to chapter 8 metabolism, chapter 9 cellular respiration, and then to start listening to this particular chapter, because this chapter is like all of these chapters are built one on another. Okay, so you have a better understanding of the topic and the subject matter. Okay, as usual, the first one or two slides, I have a list of things that you should know about this particular chapter, and it's a good idea to always quiz yourself after listening whether you know these topics or not. Okay, so once again we go back to the hill. Okay, so what I like about the hill is it gives us a very good idea, like a summation of what is involved. Okay, so in the previous chapter, the chapter on cellular respiration, we were talking about the water on top of the hill. Now, water on top of the hill is likely to have more potential energy and it has the ability to flow down. Likewise, you have glucose, okay, which is a high energy complex molecule, which when combined with oxygen in the cellular setup is likely to release energy. The whole process is exothermic, and if you actually calculate, you can get 38 ATP out of each molecule of glucose, and this process is a cumulative action of oxidative phosphorylation and substrate level phosphorylation together, and that's how you end up getting this ATP. The whole process is spontaneous, meaning it has a negative delta G. Okay, so and then it happens in the cell cytoplasm and then in the mitochondria, in different parts of the mitochondria. All of the action can be summed up in 3 steps: glycolysis, which takes place in the cytoplasm; Krebs cycle, which takes place in the mitochondrial matrix, okay, and the oxidative phosphorylation, which takes place in the inner membrane of the mitochondria, this inner membrane that is present. Okay, and at the end of it, it is completely broken down into carbon dioxide and water. These are very simple, low energy, least complex kind of molecules. Okay, on the other hand, when we talk about photosynthesis, which is what this chapter is about, in that you have first this carbon dioxide and this water being put together in a cellular setup. Okay, and that cellular setup is a plant cell which contains the chloroplast. Okay, there are other bacteria which are capable of doing the similar kind of photosynthesis, but for our course we are looking at the photosynthetic apparatus or machinery and the workings of a plant cell and the chloroplast. Okay, so you have these two molecules, simple molecules, carbon dioxide and water, which are very simple, low energy ones. They are put together. To put it together, you need energy, and that energy is obtained in the form of ATP from the sun's light energy. Okay, and you also need reducing power. What I mean by that is someone who can give protons, and then we can further manipulate this carbon dioxide molecule to give us a highly reduced complex molecule, which is our glucose. Okay, now the energy comes in the form of sun's light energy, and then it is converted into a usable form, and also reducing power is somehow managed. We sort of manage. Now the whole reaction is endothermic. It takes place in the chloroplast. It is summed up in two main steps. One is the light reaction. Okay, it is also called non-cyclic photophosphorylation, which takes place in the thylakoid membrane, and the dark reaction, which is also called the Calvin's cycle or the Calvin-Benson cycle, which takes place in the stroma liquid, this liquid part of the chloroplast. Okay, so so first we start with what is this. I normally start my class by asking my students about what this is, and everyone tells me this is a rainbow. And okay, so then you understand that when you get a rainbow, you simply assume it is the sun's light falling. You have sunlight and you have water which is falling down. So these water molecules act like prisms, and the sun's light, the visible range of that light, is split into its individual colors. Roy-G. Biv, which is red. Oh, can I go back? Just a second. Which is, okay, so your red, then orange, then yellow, then green, then blue, then indigo, and then violet. Roy-G. Biv. Okay. Okay. So the nature of sunlight is it emits light, all right, and it is a form of electromagnetic energy, which is also called the electromagnetic radiation. It has a varied range of wavelengths. Now, what is a wavelength? A wavelength, people understand it as kind of something like this that they talk about in the graph that they have encountered. So you have something like a crest and a trough. This was in physics long ago. Anyway, so the distance between one crest and one trough, that measures as the wavelength. It is represented in the form of lambda, the Greek symbol. It can range from nanometers to a kilometer in wavelength. Okay. Okay, so we move to the next slide, wherein the entire electromagnetic spectrum is the entire range of the electromagnetic energy or radiation that is emitted from sun. It has different parts to it. It can have things like, things like gamma rays, X-rays, UV rays, then visible range, then infrared, then you move on to have radio waves, microwaves, etc. Okay, we are interested in the visible. Okay, so you have the visible range split into its individual components, where you have your Roy-G. Biv. Okay, so it is from the range of 380 nanometers to 750 nanometers. Okay. Now, this is another part which is an interesting part, where what you need to understand is how do you classify different living creatures. You classify them according to their ability or what is the source of carbon that they eat or need or use, and what is their source of energy. Like for example, if you talk about, you start, okay, do you fix carbon? No, I don't fix carbon. Okay, do I get energy from light? No, I don't get energy from light. Do I do inorganic oxidation? No, I don't do inorganic oxidation. What do I use? I use organic carbon, which is my source of carbon, that is my glucose or any other carbon molecule, and my organic carbon or organic compound or a chemical is my source of energy. Okay, so I am a heterotroph, which I can also be called as a chemo organo excuse me troph. In some cases, we are just loosely classified as chemoheterotrophs. Ideally, in this particular example, this chemoheterotroph would be an organism which can perform some inorganic oxidation, kind of like use a sulfate and convert it to hydrogen sulfide, or a nitrate and convert it to ammonia, that kind of organic or inorganic oxidation processes. There can be many others also that is possible, which would be under the group of microorganisms which can do this. This is just one set of examples. There is numerous kinds of them. Okay, so that is one of the things. On the other hand, if we are talking about a plant, does it fix carbon? Yes. Does it need energy from light? Yes. So it is a photoautotroph. Anything else, a photoheterotroph, a chemoautotroph, and any other kind of autotroph, or a chemoheterotroph which does inorganic oxidation, all of these are microbes, and we are not really concerned with that for our course in this particular case. Okay, so what we need to know, or you need to know for this course, is what is an autotroph, of which what is a photoautotroph and what is a heterotroph, and we come under the bracket of chemoheterotroph. I know you would be thinking, in the previous page chemoheterotroph showed chemical, organic and inorganic oxidation, but sometimes we are bracketed under this as a general, and it is not really split into two different use of chemicals as a source of energy, and that's how they put it sometimes, and we would come under that bracket. Okay, then now when we are talking about photosynthetic pigments, the only color that comes to mind is green. Okay, now the green that comes to mind is because of the chlorophyll, because that is the only color that is reflected, that is the only wavelength that is reflected. Going back, when light falls on an object, okay, so like you have a test tube of some solution and light falls on it, what are the three consequences? Three consequences. Okay, one, you might have transmission. Two, you might have absorption. Three, you might have some light being reflected off. So the color that you see is whatever color is not absorbed or transmitted but reflected out. So that is why when you look at plants and they look green, that is because the wavelength of green range is reflected out. Okay. Chlorophyll, now plants have two types of chlorophyll. One is chlorophyll A, and this absorbs in the blue, violet, and red, and it looks blue-green when you extract. You will be doing one of these labs where you would extract chlorophyll, and you can see that it has two sets of colors, a slightly bluish kind of blue-green kind of color, or an olive green kind of color, or a very bright kind of green color. Okay, so you have chlorophyll A and chlorophyll B doing the job of photosynthesis. But there is one thing that you do not know. There are other colors which you see during fall. Why do you see during fall? Let's talk about that a little bit. One, this is just before winter. During winter, it's going to be cold. It's going to be not having water, the temperature not being conducive. So it makes more sense for the plant to rest, which means it does not do any photosynthesis. It does not lose the water that it has. So the best way a plant can protect itself in such a situation is to lose the leaf. And that is your fall. Just before it loses the leaf, it first loses the chlorophyll pigment. Once it loses the chlorophyll pigment, you can see the other pigments that are present underneath. You can have red, brown, orange. You can have rust. Then you can have yellow. You can have varied shades of these. All of these come under the bracket of being carotenoids. Now, carotenoids, what is their main purpose? They offer photoprotection. What is the meaning of photoprotection? They absorb excess light. Okay? They absorb the excess light and prevent the leaves from getting damaged because of reactions that come with excess light. Okay? What are the advantages of photosynthesis? How does photosynthesis help us in the planet Earth? Okay. One, you have plants like plants, your land plants. Then you have your algae, which are in the water, and certain prokaryotes, which are the cyanobacteria. These convert light energy and form chemical energy, and they store it in the form of a sugar. They make it out of carbon dioxide and water, which is your principle behind what is photosynthesis. Lots and lots and lots of algae nowadays can be used to grow. All you need is a tank full of water and good sunlight, and you can have algae growing. Once you get this algae, you can harvest this algae and extract the oil and use it as biodiesel, or you use those carbohydrates to generate ethanol, which is your clean fuel. Okay. Now, where does photosynthesis take place? I mean, obviously everyone says it's in the leaves, but where exactly it takes place? For that, you have to understand the makeup of the leaf. Think about it. If this is my, one second, let me just go back. Okay. If this is my leaf, so this was my leaf. Imagine it has been cut kind of like this, so you can sort of see it in this fashion. So the topmost layer and the bottommost layer, okay, they have certain things. Now the topmost layer, what does it have? It has a lot of epidermal cells, which is what these guys are, epidermis. The bottom part also has epidermal cells, but from time to time there are openings, and these openings are called stoma. A lot of them together are called stomata. Okay. Now stomata or stoma, what is it? It's like an opening to the outside. What is their main purpose? One, you have studied about the transpiration, which is a feature that happens in plants. That means water moves up from the roots to the leaves via the column, and that is done by the property of cohesion. You've heard of that, and adhesion, right? Now, there has to be an opening on the outside, and this is the opening. So then the water is lost through these openings out, and fresh water keeps coming up as a column, up constantly. So you have the stomata as an opening to let out water. Also, it has a double purpose. Carbon dioxide can go in and can meet with the cellular apparatus, which is capable of putting it together and making sugar out of it. And oxygen is released, which is like a byproduct of photosynthesis. It is released in this process. Okay, the cells in which this takes place is called the mesophyll cells. You have two different layers, tightly packed layer and a slightly looser layer. Okay. Okay. Each chloroplast is a double membrane, meaning it has got two membranes. So kind of like this. Okay. So once it has got that, be aware that it has these stacks like, a stack of pennies being arranged. So this is a stack and it is called a grana. Okay, singular is granum, and this gives a grainy appearance to the entire organelle. Okay, and it has got a membrane, and each unit is called a thylakoid. It has got a membrane, and that membrane is where the action takes place, and there is a space in between that is also called the lumen. The empty liquid part, okay, without any grana, is called the stroma. Okay. Now, photosynthesis is a redox reaction, meaning there is reduction of something, oxidation of some other things. Okay. So what happens is carbon dioxide, which is completely oxidized, gets reduced, and water, which is completely reduced, gets oxidized, okay, into oxygen. Okay, the pigments in a chloroplast absorb the photons, capturing solar power. Now, to explain this a little more, long ago when I was dealing with chapter 2 with you guys, I had told you about any time there are atoms, they have electrons moving in their orbitals. The electrons in the outermost orbital are highly unstable, okay, and when they get energized by form of heat energy, light energy, whatever energy that they encounter, they move to a very unstable stage, in a high energy unstable stage. Okay, kind of imagine if these are your, so this is an electron and it moves out of its orbit, so it has gained energy and it is moved out of its orbit. The reason why, because it gained some energy, or okay, now this is very unstable, it will fall back. Whatever goes up has to come back. When it comes back, it gives away that energy. That energy can be given away in the form of heat by some, or in the form of light by others. This is what you feel when you touch the bonnet of your car on a hot day, and you can sort of, when you walk towards it, you can feel heat emanating from that. Okay, you can feel that your car has really heated up. That is because all of those electrons that got excited went out of their orbital, came back, and when they came back, they gave away that energy that they absorbed, and that is what you feel as your heat radiating. Chlorophyll molecule, on the other hand, behaves in a slightly different manner. It does get excited. It does have its electrons getting excited and leaving the orbital and going out. When they come back, they emit light, that energy, whatever they got, in the form of light. And that is what is fluorescence. Now you might say, why is this fluorescence not happening in my normal leaf? That is because there is another sequence of reactions that is destined for this electron that went out of the orbit. So in the lab, when you have extracted chlorophyll, put it into a tube and then expose it to light, and then you take it and then you keep it in the dark or you expose it to UV, it will fluoresce. The reason is it is not inside a leaf. Now, why? A leaf has a system to deal with that electron in a different way. It will not get heated up and emanate. It has another course of action destined for this whole process, and we will see what happens. But in the lab, when you just extract chlorophyll and put it into a conical flask and expose it, that pigment is now going to fluoresce, right? Okay, so now we go on to the process of light reaction, wherein solar energy is tapped and converted into chemical energy. We are going to look at the way in which our leaf is going to do this. Okay. Okay. Just a sec. Okay. Now, excuse me. So, within the thylakoid membrane, you have a photosystem. Now, it has got a lot of other pigments and proteins. They are all organized to deal with this electron that has got energized. For simplicity, I am going to deal with the photosystems and only talk about chlorophyll molecule or what happens to the chlorophyll molecule. You have to presume that it is a combination of other pigments and the proteins and the chlorophyll that is giving you that outcome. But for simplicity's sake, I am only going to talk about it in terms of chlorophyll. Okay? So here I have, let's first be mindful, all of this reaction is taking place in the thylakoid membrane. All of this is the thylakoid membrane that we are dealing with. Okay, so there is the sun here and there is a photosystem 2. You might say, why is it called photosystem 2? Because that was the order in which it was discovered, okay, by scientists. So photosystem 1 was discovered first and photosystem 2 was discovered later. Photosystem 1 is observed in lowly bacteria and lower forms. Our plants are more evolved creatures, more sophisticated system of deriving energy, and so they have a different mechanism set up. Okay, so photosystem 2 has the ability of absorbing chlorophyll at absorbing light at 680 nanometers. Okay, when it absorbs the light energy from the chlorophyll molecule, an electron gets excited and it leaves the molecule and leaves an electron hole. Okay, so for a billionth of a second, it leaves the hole and goes out into the orbit. Now, in our lab, it came back and it fluoresced. In our real life, a live plant, that is not the way it works. What happens is it cannot come back. Why? Something else takes its place. We will deal with this later. Please hold on to the idea that that particular hole is filled. So the electron cannot come back. Okay? Where does this electron go? It goes on the membrane. There are different electron carriers. So there is like an electron carrier 1, 2, 3, 4, 5, 6, 7, many. For our simplicity, I'm just listing out them as 1, 2, 3, 4. You don't have to remember individual names. So it is taken up by the first electron carrier. The minute the electron carrier takes that up, the carrier becomes reduced because it has gained an electron. And then it passes it on to the next carrier. During this process, it is capable of taking a proton from the stroma and throws it inside into the lumen of the thylakoid. Likewise, I have this second electron carrier transferring, and again another proton thrown in, a proton thrown in, a proton thrown in. So at the end of this process, I have a lot of protons thrown inside. I have a lot of protons in the inside as compared to the stroma liquid. And then everything is so sequentially synchronized, very well synchronized, that the electron finally fills another hole. Why? What has happened? While this route is happening, another wavelength of light has hit PS1, that is photosystem 1, which is 700 nanometers, and that has excited the second chlorophyll molecule. It has created a hole and gotten excited and gone out into space. Then this hole is completed or filled in by the electrons coming from the first sequence, the transport chain that happened. Okay. And this electron now does not have a place to come back. Where does it go? It goes through a sequence of electron carriers. Okay. And finally, it reaches NADP. NADP. This is like a cousin of our NAD that we had come across in cellular respiration. Nicotinamide adenine dinucleotide. This is nicotinamide adenine dinucleotide phosphate. This is specially seen in plants. Okay, during photosynthesis it bears stronger mention. Okay, so this NADP accepts this electron along with the protons and becomes NADPH in the stroma. Now, at the end of this process, what has happened? Let's just take a recap. Light hit the first photosystem 2, created an electron hole. I had a sequence of electron carriers forming a gradient of protons inside the lumen. I had a second wavelength of light hit my photosystem 1 and excite an electron which goes through a sequence, a shorter sequence, and reaches NADP, which gets converted to NADPH. So I have reducing power in the form of NADPH. And this gradient what happens is it manages to rush out into the stroma, where in the pictures it's not really mentioned, shown, there is an ATP synthase channel protein which is conveniently placed and allows for the flowing of the protons and helps in formation of ATP from ADP and inorganic phosphate in the stroma. Okay? Okay. So let's just go over it bit by bit. Okay. First, we have photosystem 2. This is what functions first. It is non-cyclic, like a straight line. There's no turning back. Okay. It is best at absorbing light of 680 nanometers. It is called P680. Then you have photosystem 1. This comes after photosystem 2. It is best at absorbing light at 700 nanometers. It is cyclic. The reaction center of chlorophyll A of photosystem 1 is called P700. Okay, so this is like a simulation of what exactly happens. If you see, light hits the photosystem 2, the electron gets excited, goes out into space. It goes through a sequence, like a mill of sequence of electron carriers, which is responsible for making ATP. In the meanwhile, while it is making the round, another light hits photosystem 1, and I have an electron hole that is filled by this guy, and this one is conveniently reaching NADP and makes NADPH. So both ATP and NADPH are made available in the stroma. That is a very important sentence, because the next set of reactions are going to take place in the stroma. Okay, so when a photon hits the pigment, energy is passed, and finally this P680 is missing an electron. It's got an electron hole. Now, remember some time back I asked you guys to hold on to who comes and fills that hole? Now we talk about that a little more. Water molecule is split by enzyme. So I have H2O splitting into H and O. Okay. So I have two H's and O. So if I have two of this, then I'll have four H's and oxygen molecule is evolved out. Got that? So this is how oxygen is released. This is the reason cyanobacteria and plants are responsible for making oxygen available in the atmosphere. And that is why we live. Okay, so each electron falls down the electron transport chain from the primary electron acceptor of photosystem 2 to photosystem 1. This creates a gradient, and then they diffuse across the membrane. Just a minute. This creates a gradient across the thylakoid membrane inside the lumen, and diffusion across the membrane drives the ATP synthesis across the membrane into the stroma. Drives the ATP synthesis. Okay, so photosystem 1 is excited with P700, which loses an electron, and then it accepts it from the photosystem 2. Okay, now this second electron that got excited, it falls down an electron transport chain to ferredoxin, which then are transferred to NADP to give me NADPH. Okay, some bacteria have photosystem 1 but not 2. So they go through a process of cyclic electron flow, and this is more primitive method. They are only responsible for protecting cells from light induced damage. What are the different products of light reactions? This is an important one to remember. I get NADPH, I get ATP. So this is my reducing power. To reduce what? To reduce carbon dioxide to give me sugar. Okay, and oxygen is evolved. The chemiosmosis at the sequence of electrons between photosystem 2 and photosystem 1, and the part of the so this is the sequence of reaction where it is shown you in the picture as compared to the picture that I have. So this is your photosystem 2 versus photosystem 1, wherein you have the ATP being formed, oxygen being evolved, NADPH being formed, and now this is available in the stroma liquid for the process of Calvin cycle to take place. Okay, so photophosphorylation. Photophosphorylation basically means you use light to phosphorylate ADP and form ATP. Okay, this is different from oxidative phosphorylation, wherein we used the oxidation reduction methods, wherein we used mitochondria, which transferred the chemical energy from the food and helped form a chemical energy of ATP. Okay, we move to the part where we are talking about the Calvin cycle, where carbon dioxide is reduced to sugar. Okay, so Calvin cycle makes sugar. So for making of that sugar, I need atmospheric carbon dioxide and water. I also need ATP and NADPH. Okay, now Calvin cycle technically does not produce glucose. What it does produce is something known as G3P, glyceraldehyde 3-phosphate. Now, if I have 2 glyceraldehyde 3-phosphate, meaning this is a 3-carbon molecule, I can put that together to give me 1 glucose. Okay. Okay, now I have an analogy to give the class. When someone is drawing something, like for example, I'm trying to draw something. So, one second, let me just go back. See, I'm trying to draw something. Okay. So, what am I drawing? So, when you look at this, you are, I don't know. What does it feel like? I'm not sure. And then you say, oh, maybe it feels like an elephant. You sort of get the picture once greater details are done. My point being, when you have this process wherein you are dealing with one carbon dioxide molecule entering the Calvin's cycle and goes through a process, you get something, but it's not clear what you get. So The second one gets into it, it's still not clear. The third carbon dioxide gets into it, then I get something which I call it a G3P. Okay, so basically the Calvin cycle has to turn 1, 2 and 3 times over for me, 3 times for me to get a 3 carbon molecule. Now, how many times does it have to turn in order for me to get a 6 carbon molecule which is glucose? The answer is I have to turn it 6 times. So 1, 2, 3 will give me one G3P. 4, 5, 6 will give me another G3P. And this can become one 6 carbon molecule which is glucose. Is that clear? So that sort of will help us understand the next page. So what he says is, to make matters easy or understandable, the textbook asks us to follow it this way. You need carbon dioxide, you need ATP, you need NADPH. To make a G3P molecule, which is a 3 carbon molecule, you need 9 ATP and 6 NADPH. That means you have already done 3 turns of the Calvin cycle. So one turn probably took 3 ATP and 2 NADPH. But I still could not see a product that I can make some sense out of. So 3 turns consumed 9 ATP and 6 NADPH. To make one glucose molecule, which is a 6 carbon molecule, I need to have 6 turns or 2 times this. Does that make sense? So I need 18 ATP and 12 NADPH. Okay? So what are the different steps? First is fixation, then reduction, then release of the G3P, and then you have to regenerate back the molecule. Now going back to that, what you have to understand, I will try to simplify this. Okay, so here is my Calvin cycle. In this entire cycle, I have a 5 carbon molecule. I have 1 carbon dioxide getting in. I'll make a 6 carbon molecule, but it's not very clear, so I have 3 of these 5 carbon molecules. It is called ribulose bisphosphate, and the enzyme that puts together 3 carbon dioxide molecules is Rubisco. This is the world's most important protein. So what do I get? I get 6 3 carbon molecules. So this is 15 plus this 3 is 18, and this is how I get it. So these are restructured. Okay, this is the first stable product. That 6 carbon that I initially get is not very stable. The first stable, something that you can make sense out of, is 3PGA or phosphoglyceraldehyde, and that is then converted to 6 of the G3P. It's glyceraldehyde 3-phosphate, of which 1 goes out and the rest of the 5 is restructured to give me ribulose bisphosphate. So if you notice, it is 6 molecules of 3PGA, which is then restructured to give me 6 molecules of G3P, that of which 1 goes out and can be used to make glucose or other compounds, and the 5 of them are restructured back into the scene to take another rounds of the next set. Okay, In short, for this whole process, I will consume 9 ATP and 6 NADPH. This is with just 3 carbon dioxide. And I have just made 1 G3P. This is not a glucose. If I want to make glucose, I need 3 more carbon dioxide to get into it. Okay? And then I can make 1 more G3P, which can become a 6 carbon glucose. Okay, so now we move on to the part where we are talking about what are the evolutionary characteristics of different scenarios. Now, this particular scenario wherein we talked about Calvin-Benson cycle, where the first stable product that we got was 3-PGA, which is a 3 carbon molecule. It is called, a plant capable of doing that is called a C3 plant. Okay? This is the first stable compound that is being made. So it is called a C3 plant. Now, think about it like this. On a hot, dry day, what will happen? It is, once again, excuse me. On a hot, dry day, it is hot. So what would you do? The stomata will close. Why? It wants to conserve water. Now, if it conserves water, what happens? Yes, water is conserved. I do not lose the water to the outside. But the stomata is closed, so I have no carbon dioxide coming in. So I don't have enough carbon dioxide. So can I do Calvin's cycle? No, I cannot. But am I stopping light reaction? No, light reaction is not stopped. So that means I do have oxygen being evolved, and that oxygen is not going out, and suddenly inside the leaf I have a huge big buildup of oxygen. I don't have carbon dioxide, but I have a huge buildup of oxygen. So what happens in such a situation? There is a lot of things that happen to it. So what does oxygen do? Oxygen combines with Rubisco enzyme plus some fixed carbon, already fixed carbon, and I make use of ATP, and I just evolve carbon dioxide. So it is like the plant had put so much energy in fixing this carbon. It had put 18 ATP, 12 NADPH in fixing the carbon, and that is just getting burnt up by oxygen because of its affinity to Rubisco enzyme. You might think it is very, very similar to burning a $500 note. If you're just burning that, what a waste, right? How much someone must have put an effort, worked to earn that money, and you're just burning it off, right? Kind of like that situation. So let's see. This is a situation that happens, okay? So what happens in that process? I can see this is what is happening in my leaf. I am using up oxygen that is made by the light reaction, and I am releasing carbon dioxide. So does it feel similar to just respiration? So it is called photorespiration because it happens in plants, but it is not respiration. Please It is not respiration. Okay, what is this? This is a waste. If it is so wasteful, why is the plant still doing it? The reason then they studied is this is the plant's way of removing the excess oxygen. Otherwise, oxygen can form a lot of radicals and destroy tissue, destroy the plant tissue. And the plant thinks to itself, yeah, this happens, but it happens only sometimes on a hot, dry day. That's all. I can deal with that. Okay. Now, the earth. Okay. This is the equator. These are your temperate regions. C3 plants are more concentrated in this region. Meaning plants which make the first product, which is the 3-carbon compound, in the form that I explained to you in these regions. What is the reason? Why am I mentioning it? Because hot and dry is not a very common situation in these parts. It's not. It's like once in a while. But mindful, be mindful. Literally 50% of the plant's fixed carbon is just lost on such days. Thankfully, it doesn't happen very frequently. It doesn't happen very, very frequently. All right. So we can go ahead and look at how the plant evolves in different regions where it is constantly hot or dry. Okay. So think about it like this. There is a spot wherein Calvin cycle is taking place, and that spot is cut off with a chamber separation to the spot where stomata is open or closed or oxygen being evolved and so on. Meaning they are trying to keep Rubisco enzyme away from oxygen and artificially create increased levels of carbon dioxide in this region, in this specific region. So Calvin cycle will take place without a problem. Okay, so C4 plants, what do? They first fix the carbon into a 4-carbon compound. So here is where I have, okay, so carbon dioxide comes in and there is a 3-carbon compound and it turns into a 4-carbon compound. This is what is let in. This releases carbon dioxide here and comes back here and then is ready to take the next carbon dioxide molecule. This way it eliminates the waste occurring when oxygen and Rubisco get to meet. They try to avoid them meeting by separating them into different chambers. Okay, so what I have is basically a situation where, okay, we'll talk about 2 different situations. Now here I have a 3-carbon compound. I have carbon dioxide into the picture. They become a 4-carbon compound. This is released. 4-carbon compound releases carbon dioxide and becomes a 3-carbon and comes back here. And this is in the Calvin's cycle. So in this particular case, the first stable product that is formed is a 4-carbon compound, and that is the reason the plants that follow this system are called C4 plants. On the other hand, an example of this is sugarcane. On the other hand, I have another situation wherein cactus. What does a cactus do? You all know cactus lives in a desert, okay? And there is no way stomata can open in the daytime. So the stomata opens only in the night. So at night, when there is the moon and the stars, the stomata open, carbon dioxide comes in, and it is fixed into an organic acid and stored like an organic acid. Okay, it is also called crassulacean acid or CAM. This releases carbon dioxide for the Calvin cycle. So the only difference between this C4 plant and this CAM plant is the timing at which they take place. So in the daytime, when there is light reaction taking place, I have an artificial increased quantity of carbon dioxide created by this organic acid releasing the carbon dioxide for Calvin cycle to take place. Okay, so hot dry day, you have CAM plants such as pineapples and cacti. They conserve water by opening their stomata and admitting carbon dioxide only at night, and that is also a 4-carbon compound. So they are also C4 plants. They bank all the carbon dioxide at night and release it during the daytime for Calvin cycle to take place. Okay, so this is like a compilation of all the 3 types. So you have in respiration, which is your cellular respiration, oxygen in, carbon dioxide out. What happens? Glucose is broken down, 38 ATP molecules are made. It takes place in the mitochondria and on an everyday basis. In photosynthesis, carbon dioxide is in, oxygen is out. Glucose is made by using 18 ATP and 12 NADPH for this process. It takes place in chloroplast on an everyday basis in the daytime. Okay, photorespiration on the other hand, you have oxygen in and carbon dioxide out by using a fixed carbon and ATP and releasing it out as carbon dioxide. This takes place only in hot dry days in C3 plants, and it drains away literally 50% of the fixed carbon from plants. There are adaptations to overcome this, and the best examples that we have come across are C4 and CAM plants. This is a compilation of the picture which sort of puts together the entire photosynthetic reaction in the chloroplast. So you have your light, your water. In your thylakoids, these two are taken in, put together in the photosystem with light reactions using photosystem 1 and 2. Releasing oxygen out is one of the outcomes, but also making ATP and NADPH and making it available in the stroma. That is taken up by the RUBP molecules and Rubisco enzyme. This is important to make 3-PGA, which then converts to give me G3P. This G3P can be put together to give me sugars, which can be used for cellular respiration, make cellulose, starch, other organic compounds, etc. Okay. So photosynthesis converts light energy and includes light reaction and dark reactions. Light energy into chemical energy. In the light reaction, water is split and oxygen is released. Okay, light excited electrons of chlorophyll are passed down the electron transport chain, producing ATP by chemiosmosis. They also reduce NADP to NADPH. Okay, and carbon dioxide is fixed to RuBP molecule, which is the 5-carbon molecule, and the NADPH is used, okay, to form G3P. Okay. This completes the chapter. Thank you.